Hi everyone, it's Nock and Haley, and today our group will talk about cellular respiration process. So first, let's find out what is cellular respiration. Cellular respiration is the process by which organisms use oxygen to break down food molecules in the form of sugar to get chemical energy for cell function. There are 4 steps in this process. First is glycolysis, pyruvate oxidation, citric acid or Krebs cycles, and the last one, oxidative phosphorylation.
The first step is glycolysis. Glycolysis occurs in the cytoplasm of the cell. Glycolysis is a term that means taking sugar and splitting it. There are 4 small steps in glycolysis. The first one is phosphorylation of glucose to glucose phosphate. First made more reactive by additions of 2 phosphate molecules, which come from hydrolysis of 2 ATP, provides energy to activate glucose and lowers activation energy. The second step is splitting of phosphorylated glucose, split into 2,3-carbon triose phosphate. The third step is oxidation of triose phosphate. Hydrogen removed from 2-triose phosphate and transfer to NAD, which means hydrogen carry coenzyme to form reduced NAD. The last step is production of ATP. Enzyme control reactions convert 2-triose phosphate into 2,3-carbon pyruvate. The process also regenerates 2 ATP from ADP plus PI. In total, glycolysis produces 2 pyruvate molecules, 2 molecules of ATP, and 2 NADH molecules, which go to the electron transport chain.
The next step is pyruvate oxidation. Pyruvate oxidation takes place in the mitochondrial matrix. Before the reaction in this process can begin, pyruvate must enter the mitochondrion, passing through its inner membrane and to the matrix. In the matrix, pyruvate is modified in a series of steps. Firstly, a carboxyl group is removed from pyruvate and released as carbon dioxide. The resulting 2 carbon molecules is oxidized and NAD+ acts as the electron acceptor for the lost electrons, forming NADH. The oxidized 2 carbon molecules is attached to coenzyme A to form acetyl-CoA. Acetyl-CoA carries the acetyl group to the Krebs cycle, which is the next step of the process. These steps are carried out by a large enzyme complex called the pyruvate dehydrogenase complex. This is a key target for regulation as it controls the amount of acetyl-CoA that can enter the Krebs cycle. For each glucose molecule, 2 molecules of pyruvate are converted into 2 molecules of acetyl-CoA during the pyruvate oxidation, releasing 2 carbons as carbon dioxide and generating 2 NADH from NAD+. Acetyl-CoA serves as fuel for the Krebs cycle in the next stage of cellular respiration.
The third step is citric acid cycle. The Krebs or citric acid cycle is the main source of energy for cells and an important part of aerobic respiration. It occurs inside the matrix of the mitochondria. The Krebs cycle begins with an enzymatic addition reaction of acetyl-CoA to oxaloacetate, forming citrate. The citrate is isomerized by a hydration sequence to yield isocitrate. Further, enzymatic oxidation and decarboxylation gives to ketoglutarate.
After another enzymatic decarboxylation and oxidation, 2-ketoglutarate is transformed into succinyl-CoA. The hydrolysis of this metabolite to succinate is coupled to the phosphorylation of guanosine diphosphate, which is GDP, to guanosine triphosphate GTP. Enzymatic desaturation by flavin adenine dinucleotide FAD dependent succinate dehydrogenase yields fumarate. After stereospecific hydration, fumarate catalyzed by fumarase is transformed to L-malate. The last step of NAD-coupled oxidation of L-malate to oxaloacetate is catalyzed by malate dehydrogenase and closes the cycle.
The last step is oxidative phosphorylation. Phosphorylation is the process of giving a substance, such sugar, a phosphate group. ATP is phosphorylated from ADP in the case of oxidative phosphorylation. Occurs on the inner mitochondrial membrane with protons diffusing across into the membrane and later come back into the matrix. Involves the destruction of the electron transporters FADH2 and NADH to produce ATP. There are about 34 ATP produced by oxidative phosphorylation. It is a catabolic process since the electron carriers are being broken down. We use the word around because the quantity of ATP produced and can vary depending on how many ions the complexes in the electron transport chain are able to pump through.
These are our hand drawing of all 4 steps in the cellular respiration process. That is the end of our presentation. Thank you for listening.